Hi, so this video is going to go over A1, so that is going to be unity and diversity of molecules. For my class, this is actually our 5th topic we're doing, but if you're watching this and going in order or reviewing them in order, this might be your starting point. So just a quick little how it's going to work. I will do 1 page per each subtopic, so there'll be 1 page of notes. I will probably end up having to erase my board a couple times, but if you want to try to fit it on 1 page, it should, in theory, fit on 1 page, though feel free to use page front and back or 2 pages, whatever works for you. This is just my little way of making sure I keep it concise. So there will be 1 page on water essentially and 1 page on DNA. The pages are fairly full, so if you are trying to keep it to 1 page, write small. Otherwise, write however you want and use whatever space you want. Okay, so we are going to go through this topic has 2 kind of fairly separate areas, so water and the nucleic acids. They're not super well related. They both have hydrogen bonds. It's probably about the extent of their connection, but they kind of pick up this unity and diversity. So the idea is that water would be the unity, the unifying thing between all living organisms is they're reliant on water, and then the diversity would come from the DNA of the nucleic acids. Different nucleic acids give rise to different features. So that's kind of, I think, how they ended up being paired together. Okay, so let's start with kind of the statement of fact, which again, where it comes into this idea of unity, that water is essential for life. So water, and particularly liquid water, is essential for life. So most things that rely on water need it to be in liquid form. So that's kind of why I drew attention to that particular aspect of it. Okay, so we're going to actually pick up on a few things from that one statement. So in terms of why is it essential, a couple of different things you would want to be able to pull out of that. So first of all, cells originated from water. So current cells kind of took water, and that was useful in terms of formulating a cytoplasm. So cells originated from water and are primarily made up of water. So cytoplasm is an aqueous solution. So anytime we see aqueous, that just means water-based. And most kind of reactions that are needed for living things take place in water. So it is the medium for living processes. So those are going to be reactions. Most enzymes actually work in aqueous environments. So most of the kind of living processes that we need require it. So I'm going to leave a bit of space to the side. So I'm going to go into a bit and then kind of come back to this. But when we say that it is required for life, we can also then question, well, where did that come from? So life on Earth is only possible because there is liquid water. So we're going to kind of clarify that. Due to the large volume of liquid water. And so then the question becomes, well, where did that come from? So where did that water come from? And therefore, can we use that to figure out where else there might be life? So we're going to come back to that after we talk a little bit more about water. That is going to be our little HL extension. That's actually the only part of this water stuff that is higher level only. So I'm going to go through all the rest, and then at the end of this section, I'll cover the HL material. So if you're SL, you don't have to kind of go through that part, but you do need the rest. Okay, so that's kind of why it's essential. Fairly short explanation on that, but that'll be more obvious as you work your way through the course and see all the different places where water shows up. But the more important thing is that you're really, really familiar with the structure of water. So if we look at kind of with the new course, why would this be 1.1? Like, why is this, in theory, the first thing you would learn? And that's because if you want to understand how our cells structured, you need to understand the role of water. If we want to look at essentially how do chemical reactions take place, we want to understand how that relates to water. So this structure of water kind of comes into all of the rest. It plays a key role in different parts of our body and so forth. So we want to kind of make sure we have a really good understanding of that because it plays an aspect in so many other parts of life. Okay, so you need to be able to draw the structure of water. So I'm drawing an oxygen with 2 hydrogens. That is 1 water molecule. And I'm drawing a covalent bond between them. So that covalent bond is it's a polar covalent bond. So we're going to clarify that. So it is a polar covalent bond. And when we say polar, what do we mean by that? So it means the electrons are unequally shared. Which creates slight charges. So if you're a chemistry person, you will understand this most likely better than this, like you will understand it at a very kind of clear level. I'm explaining the level you need to understand it as a biology student. So you just need unequal sharing, so you can kind of learn some of those terms. So term is unequal sharing. So the unequal sharing actually pulls the electrons closer to the oxygen. So that's going to give it this little slight negative charge. And then because the electrons are pulled away from the hydrogen, it's going to result in a slight positive charge on the hydrogens. So you only need to use the term unequal sharing if you want to kind of add to that pulled towards the oxygen. That might help you remember why the oxygen is the one with the slightly negative charge. So that results in kind of slight charges. The term for this result of the slight charges is polarity. So anytime you are talking about water, you want to first say polarity. So you want to, that's kind of always your starting point. So everything else comes from that. So if you're talking water, you are starting with polarity. So then we're going to look at kind of consequences of polarity and kind of look at them as kind of 2 categories. We'll end up going all the way down to physical properties, which actually are still related to the polarity. So essentially, no matter what you write about, the polarity is playing some role in it. So let's say, what are some consequences of the polarity of water? So for now, I'm going to look at it as kind of 2 categories. So one of it relates to its ability to form hydrogen bonds with other polar substances, and the other relates to essentially its solvent properties. So I am going to kind of establish those separately. So the consequences, so both of these are kind of consequences. One is it forms hydrogen bonds with, and then we're going to look at that separately. So it forms hydrogen bonds with other polar things, and that includes other water molecules. And that's probably the one that's going to come up the most, because if water is polar, then the other water molecules are often also polar. And then the other is that it forms hydrogen molecules with other polar substances. So not water, but other things that are also polar, it has the ability to form hydrogen bonds with. So those are the 2 similar things. They're both related to its polarity, slight difference in terms of what it bonds to. And then the other consequence of that is going to be that it actually is a great solvent, so makes an excellent solvent. And again, leaving that little bit of space over there, just because we are going to add our HL material to that part kind of at the end. So it makes an excellent solvent. We'll do this first because this is a slightly smaller point. Then we'll go down into that. So when we say it makes an excellent solvent, it means it dissolves other materials. So it is effective at dissolving many different things. The two main things it dissolves is going to be ionic substances. So ionic substances, because they also have charges, so it's going to split your sodium and your chloride, and your salt, your sodium that's positive is going to attach to the oxygens. Your chloride that's negative is going to attach to the hydrogens. So it's able to kind of split ionic substances. It also dissolves other polar substances. And that would be, for example, like glucose. So sugars, things like that, also dissolve in water. Small non-polar things like gases can also dissolve in water if they're really small. So that's just something that's going to become important when we do cell transport later. But for right now, we're really talking more about their ability to really split things. And so when these things that can dissolve, anything that's ionic or polar, we call that, both of these, we call that hydrophilic, and that is just the fancy term for dissolves in water. So things that are hydrophilic are attracted to water and therefore will dissolve in that medium. So hydrophilic, and then you can think of that as dissolves or attraction. So attracted to water. It is important to note that we say it's an excellent solvent, not a universal solvent. So be careful of that terminology because we do want to make a note that non-polar, actually I'm going to change that word, sorry. I'm just going to say it does not dissolve hydrophobic substances. So some non-polar things, if they're really small, can actually dissolve in it, like oxygen and carbon dioxide. It's really important. But in general, it does not dissolve hydrophobic molecules. And the best example of that is going to be lipids. We're going to get to so much more of that when we get to cell membranes, steroid hormones, things like that. So this is just a little intro to that. There'll be a lot more discussion of that kind of as we go. So that's kind of one little separate point, is that we understand that because of the charges, it's able to dissolve things that are also charged, whether it's ionic or polar. Okay, so then forms hydrogen bonds with either other water molecules or other substances, and you do need to know both of these terms. So if it is with other polar substances, we call that cohesion. And if it is, if it's with other water molecules, so water to water is cohesion, co equals same molecule. If it's to other polar substances, we call that adhesion. So to stick to something, like an adhesive. Cool, so I'm actually going to, at this point, I'm going to actually leave this. I'm going to erase and move that up because I want to kind of be able to keep the flow. So I want to say like this creates this, and I'm running out of space a little bit. So pause for 1 second. You don't need to erase because you don't have a whiteboard. But I'm just going to move this up higher just so we can have the space to kind of keep the flow going. I'm going to leave that there to remind us to come back to it. Okay, so where we left off with is two other water molecules. And then, so this is like to be continued from below. So I just kept the title there. And then two other polar substances. And then we defined those as, sorry, you don't need to write this again, you already have it. I'm just recreating it because it's important for what I'm going to do next. As cohesion, and this as adhesion. Okay, so then the next thing, in order to be able to account for kind of cohesion, you would need to be able to draw it. And you need to be really clear that you know exactly where the bonds are between what things. So you need to be really good at either drawing and explaining. So both of those two things. So if we were drawing cohesion, we would say O H, like this. And I go slight negative, slight positive, and then making sure that we show that it's to a different water molecule. So here is the attraction is between the positive of one hydrogen and the oxygen of another molecule. So there's where the attraction is. So that's where the hydrogen bond forms. So it forms right there. And you could draw more than that. You could keep going. I could draw a different one over here. So I could do another, and it can keep going from there. Slight negative, slight positive, slight positive. Could actually end up forming 4, but you don't need that level of detail. Just that it is between two different water molecules and what is attracted to what. So you would need to be able to make sure you could put that in words as well. So if you were putting it in words, you would want to say and you can phrase it either way. So you can say the slight positive of one hydrogen atom is attracted to the slight negative of the oxygen, and this is going to be really key right now, of a different water molecule. So that's really what you want to be clear on, is if we're talking that, it's not talking between the hydrogen and oxygen of the same one. That's just a polar covalent bond. And then a weak hydrogen bond forms between them. And you also could have just gone straight, instead of saying attraction, said a weak hydrogen bond forms between the hydrogen atom and that of another molecule. So make sure that's kind of your key phrase of a different water molecule, that you're really good on that, because that will be important if you're going to explain it. Okay, so then if we're going to keep going, there's some implications of that. So these hydrogen cohesion bonds create two things. So there's kind of two impacts of that you need to know. So one is going to be just kind of by itself, and then the other one is going to be with them together. So if we say cohesion plus adhesion together creates capillary action. So these are kind of the two consequences you need to be able to account for. So we have surface tension and capillary action. And both of these are related. So surface tension is really only related to cohesion. Capillary action does benefit from both of them. So if we say just quickly, what are they? So if we're talking surface tension, we are saying at the surface, what you find is that they're usually bonded kind of both up and down and to the side. And so all of the bonds are a little bit weaker. But if you don't have a bond above, it actually bonds stronger to the ones next to it. So it'd be the argument, if you have like 40 friends, there's only so strong your friendships can be because you got too many friends. If you only have 3 friends, they're probably really strong. Silly analogy, but the idea is the more things it bonds to, the weaker the bonds. So the ones at the surface form fewer hydrogen bonds, and therefore they're stronger. So stronger hydrogen bonds at the surface water molecules. And you don't necessarily have to be able to go into a long explanation of that. Basically, you just have to be able to say there is those hydrogen bonds, and then they are particularly strong right at the surface. And then the result of that is it basically creates almost like a solid. So it creates some tension or some strength. And a benefit of that is some animals and insects can actually even walk across it. So one adaptation is that some insects actually walk on water. So that's just kind of what is the biological significance of that. Okay, so then let's go from capillary action. So capillary action is going to rely on both. So capillary action, what is it? Is the movement of water against gravity in thin tubes. So if the tube is really thin, water can actually move up. So it's used in like paper chromatography, it's used in paper towels, that's how they absorb. So there's lots of examples of that. The specific example that you need to be able to account for is the role of this in plant transport. So in plants, the thin tube is called xylem. And so what actually happens in plants is that water is pulled out of the leaves, and then as water is pulled out of the leaves, the cohesion pulls each water molecule below it up. So it actually pulls water molecules. So cohesion pulls H2O molecules up towards the leaves. And again, that's going to be against gravity. And so the idea is it's creating this pull. So it's cohesion, but the kind of key factor is as one molecule leaves the leaf that pulls the next one. So kind of fancy term for that is transpiration pull. And this one is going to be more, once we've done more on plants and transport, this will fill in a little bit more for you. But essentially the idea is it's trying to go out the leaves. So as one water molecule goes out the leaves, I'm trying to see if I have a little bit of room here. So here's my leaf and here's my stomata. And then as this water molecule exits, what it does is it pulls all of the ones below it because they were all holding hands, so to speak. So it's going to pull all of them up and out the leaf. So that's cohesion. It pulls one molecules towards the leaf so that as one exits, it's pulling the other. It also helps because of adhesion. So adhesion also aids. And particularly its adhesion to the polar cell walls of the xylem. Helps pull them up. So again, anytime we talk cohesion, it's the molecule, the actual between these. So this is all the connections between the water molecules. But when we actually look at that as existing inside of xylem, then the water molecules are also aided by kind of grabbing onto the sides. So that kind of enables them to kind of pull up. And in fact, even the cell wall of that leaf kind of has an extra pull. So it's pulled to the water, but also pulled to the actual cell wall of the leaf. So that aids in the pull as well. So adhesion and cohesion together kind of cause it to continuously pull up. And so the adhesion is to the cell walls, the cohesion is to other water molecules. So you need to be able to, for now, account for it really well in terms of what role each of those are. So the idea that cohesion is the pull to other water molecules is what pulls it up, whereas the adhesion is the kind of pull towards the cell walls that pulls it up, and that they both together create that movement against gravity. And remember, if you are writing about that, you always have to kind of go the whole way. So polarity creates hydrogen bonding potential, such as cohesion and adhesion, and that's what creates it. So always link it back to the polarity so that you're kind of going all the way through. Okay, so I'm going to I'm gonna go slightly out of order just because I want to erase all of that and then do the table at the bottom. So this little section here is HL only. So right now for my class, we're all doing all of it. So you're hanging in there. If you're rewatching this at some point or using as revision in your SL, just tune out for a minute. It's not very hard, but you don't need to know this. So this is the little bit of HL only material, which is our fun little like space little aspect. So we're saying life on Earth is only possible due to the large volume of liquid H2O. So then the question becomes, where did that large volume of liquid water come from? And there's multiple theories, but you only have to know one. So this is not like you have to compare and contrast theories. You have to know exactly one theory. So the theory that you have to know is the extraplanetary origin. So I'm just telling you that term because that's what they put in your syllabus. So I want to make sure if you see a question that says like, explain an extraplanetary origin, you're like, what? It's just asteroids. So that's it. That's all you have to know. So basically the one theory that you need to be able to say is that asteroids are hydrated rock. So what we're saying by hydrated rock is essentially there's lots of water trapped in the rock molecules. And when it collides with Earth, it's basically splatters. It like breaks apart into a million pieces, like if I dropped a plate on the ground, and then that frees all the water. So it collides, so the hydrated rock that release H2O when they collided with Earth, and in particular with early Earth. There's a couple different kind of evidences for that. One is that there is evidence of a lot of large asteroid collisions around the time of the formation of Earth, so kind of the timeline lines up. And then there's also evidence with looking at that idea of heavy water, so the water that has the extra neutron, the duodenum, or whatever it's called. It's in your example one in the Oxford book, has a practice question on it, but that word is not in your syllabus, so you don't need to be able to like naturally explain that. You might just have to interpret data or something on it, but essentially the like composition of water is quite similar to the composition of asteroids. So asteroids have their water looks similar to the water on Earth in terms of its kind of specific qualities of molecular qualities. So that's kind of the evidence of why we think that might be the case. So that's where did it come from? The next question that we need to be able to ask is, why did it stay? So how was it retained? So when we say retained, we're saying like, why did it not just go away? So we know that the asteroid brought it, and now we're asking, okay, why did it stay around? And there's two factors you have to be able to explain for that. So factor one is gravity. So essentially because of the size of Earth, we have a large force of gravity. So large force of gravity, which is due to our size, holds the water close to the surface. So if you don't have enough gravity, then the water lands on Earth, but then just kind of goes back out into the solar system. So this kind of gravity force is keeping it close. And then the second factor is the temperature. So it's really important for it to stay in liquid form. So for that to happen, you have to have just the right temperature. So the temperature keeps water largely in liquid form. So of course we do have at the poles ice, but the majority of Earth is actually in liquid form, and it never boils. So there is no place on Earth where it boils. There are a few places where it freezes, but in general it is staying in liquid form. So that is really important. So if you're going to have life, that has to be the case. So you have to have that just right temperature. So the temperature must be just right for liquid water. And then you need to know that kind of funny little link, that idea of just right is called the Goldilocks zone. It's also called the habitable zone. But because your book set calls it the Goldilocks zone, know it that way, which just basically means it can't be too close or too far from a star. So it cannot be too close, then it would be too hot, or too far, in which case it would be too cold, from a star, in our case, the sun. So then the implication of that is if we were looking for extraterrestrial life, so are there other places than Earth where there might be life, we would only look in a Goldilocks zone. So you would only search for extraterrestrial life in a Goldilocks zone. So if you're wanting to find ET, you better be looking in a Goldilocks zone. So looking for extraterrestrial life in either our Goldilocks zone, or can be any Goldilocks zone within kind of as long as it's within its own star. So it doesn't have to be the sun, but within the star. So that's the little bit of HL only material to add. So it's just those little bit, it's not hard, but you just have to know those extra couple of terms really. Okay, so then last thing we're gonna do, and I'm down to kind of my last maybe quarter of the page. So again, if you've written too large and you want to put this on the next page, do it. If you've only filled up half your page, then you can make a really giant diagram box now. So we are going to do a table looking at the physical properties of water. So I'm actually going to erase all of this so we kind of have some more space. So physical properties of water, so everything we've looked at before is kind of specifically about its chemical bonding. So the actual cohesion, adhesion, and surface tension and all that. Now we're looking at kind of impacts on like water as a substance. And so like a pool of water, what properties does that have? And in particular, in comparison to air is how the course has phrased it. So we're gonna kind of do our table accordingly. All right, race. Okay, so again, I want to make sure, in case you were writing like a longer response on this, that we don't lose sight of the fact that this is all due to hydrogen bonding still. So we're going to say polarity to cohesion causes these. I would give you a general advice is that they seem to really like hydrogen bonds. So if you are saying cohesion, make sure you define it as the hydrogen bonds between water molecules. So don't use the term cohesion without an explanation for it. So say the cohesion, which is hydrogen bonds between water molecules. So just be careful because sometimes they won't give points for cohesion, but they will for hydrogen bonding between molecules. So when in doubt, say both. So this is going to cause the physical properties of water. And this can go across the whole distance of your sheet because we're no longer saving that space. Okay, so we're going to make a little table. We're going to say the property. Then we're going to give a definition of that property. Then we're going to state how that property compares between water and air. And spoiler alert, it's always higher in water because of the hydrogen bonds. But we're going to write it for each one. And then in your course, there's two named examples. So I try to, anytime it gives a named example, go through it. And so the two you need to know are the loon. The loon is a duck, if you don't know the name loon, because I certainly didn't. And then you need to know a link to a seal. So however much space you have on your sheet, that's it. That takes up the whole one. And I probably should have spaced it a little bit differently, but that's all right. So property, definition, water versus air, links to the duck, links to the seal. And then there's 4 properties you need to be able to do this for. So the first property is buoyancy. So then what is buoyancy? So buoyancy is the upwards force on a placed object. So upwards force on a placed object. So if an object is kind of placed on it, is there kind of an opposite force to gravity? So gravity is the downwards force. What is the upwards force? Air has very little buoyancy. Water, by comparison, does have buoyancy, so it's considerably higher for water. So it's actually pretty high for water and higher than air. So high for water, low for air. Okay, and then how does that help the duck? So we'll have it's gonna be my duck color. So my duck uses that to float across the surface. So ducks kind of sit on water and they actually use buoyancy to enable them to do that. Seals are a little bit larger, so they have a little bit of a harder time floating on the surface, but they do use it to keep their snout above water. So the rest of them can go under, but they're able to kind of float enough that keeps their snout above water, and that's really helpful for breathing. So that enables them to kind of continually access atmospheric oxygen. Okay, so there's an example. Buoyancy is the trait. There's your definition. There's how it compares. There's your link. Next one is viscosity. And a good way of thinking of viscosity is it's kind of the thickness, which is kind of a strange way of thinking about it. The way we're really looking at it here is essentially the resistance to movement. So how easy is it to move through it? So it's not high for water, but it is very low for air. So there's almost none, so basically non-existent for air. So there's a little bit of air kind of resistance. You get a small amount, particularly if you go at enough kind of speed, but very, very low for air. And then it is low but higher for water. So water doesn't necessarily have a particularly high viscosity if you compare it to other liquids, but compared to air. So again, if you walk through the air, you would reach no resistance, but if you're swimming, we use muscle strength. There's resistance at moving through it. So that's kind of the example, how to think of it. So if ducks are moving along the surface, they do experience that resistance. So they have webbed feet and a streamlined shape. I don't really know what a streamlined shape means, but that is your official words that they use in the book. So it just means like thin shape, I don't know. A streamlined shape to move through the water. So they kind of float on the surface, but they are gonna have to kind of move from one area to another. So under the surface, they're using their little webbed feet to kind of push through because it's harder to move in water than it is to move on land. And similar for the seal, so they also have good feet, so paddle feet. And again, they have allegedly a nice shape, and that is to aid in swimming through water. So swimming takes more energy than walking, so they have some structures that kind of help them with overcoming that viscosity because it is higher for water. Okay, last two are going to be kind of thermal properties. So thermal conductivity. So this is essentially a measure of how much does it steal heat from an organism or from a surface. So it is the rate at which it transfers and passes heat through it. So the rate that heat transfers to or passes through. So you can kind of think of it to what extent does it steal heat. It's not quite like the right definition, but it is certainly how we're going to look at it for these organisms. And again, because this is always the answer, it is higher for water. These two in particular are both very clearly linked to the H bonds. So you can kind of very clearly link those two hydrogen bonds. The heat capacities are somewhat related. So they are related to the kind of stronger bonding, but it's a little bit more complex in terms of the physics behind it. So these two are kind of more evenly linked directly to that. So again, higher for water, and so as a result, if they are in water, they are at risk of water stealing their heat, so they have adaptations to prevent that. So the duck has an oil gland, and it applies oil over its feathers to waterproof them, so that way they're not having the water touch their feathers and steal the heat from their feathers. So it keeps the water away. For the seal, they are not waterproofing, so they're going to lose heat. So they have a thick layer of blubber. And that's going to help because to compensate for heat loss. So that would be like if you went swimming and you wore a wetsuit. So you were aware that the water is cold and will grab your heat. So you're going to put something into place to try to compensate or trap more heat to kind of overcome that. So that's what they're going to do. And then a last one. And again, this kind of relates to hydrogen bonding, making it more difficult to change state and so forth. So this is the specific heat capacity. So this is related not to moving heat, but how much heat is needed to actually change the temperature. So water is fairly resistant to temperature changes and state changes. So it requires quite a lot of change. And that's related to its stability in liquid form. Because of the hydrogen bonding, it's quite difficult to change its state and even change its temperature. And so this is the amount of heat or energy needed to increase the temperature by 1 degree. And if you think about this, this should be fairly obvious. It starts getting warm in summer. It feels very warm on a warm day in the outside air, but then you go in the water and it's still very cold. So water heats up much slower than air. So water changes temp slower than air. What that actually means is that H2O has a higher specific heat. So for all of these, when in doubt, it's higher for H2O because that is always the answer. Just trying to make sure you also understand what that means. So in both cases, it basically depends on the extreme. So living near water actually helps. So the closeness to water can help them maintain proximity to water to maintain body temp because of its more stability. So if you live near water, it's less prone to kind of rapid shifts. So that actually helps them maintain their body temperature compared to kind of a constantly changing environment. And then to the seal, kind of similar idea, but particularly kind of in the colder environment. So protected by cold Arctic air, by the comparatively warmer water, kind of at the start of seasons. So it's kind of a weird one because it's obviously going to do the opposite. When it then starts getting warmer, the water is going to stay colder. So that one just kind of depends. It can be a blessing and a curse, so they just kind of have to kind of modulate that. But if they have the option of living sometimes on land and sometimes on water, they can pick the environment that is the more ideal temperature. Okay, so that's all things A1.1. I'm going to erase and then restart A1.2. Okay, moving on to nucleic acids. So again, this is going to all fit in one page. If you're happy to write fairly small, the diagram's relatively tiny. If you don't want to have to worry about writing small, then just plan to use 2 sheets of paper. But if you like the idea of trying to maintain 1 page per subtopic, it can fit. Okay, so, and there's going to be a little bit of there's 3 HL things, 3 or 4 that we need to, 4 I think that we need to allocate, but they kind of mix in. So I'll mark where it's HL material only, just so you know, but we'll kind of embed it in throughout. Okay, so let's start with, again, where does this come into diversity? Well, DNA is different between different organisms because that's actually the source of the genetic code and what gives instructions. So I want to actually start with the purpose of nucleic acids. So the purpose of nucleic acids is involved in genetic information or the instructions for heritable traits. So we're going to write it as both. So involved in genetic information, but if you're like, okay, well, what does that actually mean by genetic information? It is kind of basically passes on instructions for heritable traits. And then kind of like last time, we're going to kind of have a little, how do we know that as an HL thing? So some of the HL material is like, okay, all good and well. You learn at SL, and then HL, the extension is like, okay, well, where's the background? How do we actually know that? So if this is the genetic material, the HL question becomes, how do we actually know that? So how do we know that it is in fact DNA that is the genetic material, is the question. So how do we know that? And the answer to that is the Hershey and Chase experiment. So I'm going to go a little out of order and go ahead and do this HL material because it kind of establishes a few things in terms of how we know all that, and then we'll kind of keep going. So slightly out of order. So if you're not an HL student and you are using this to revise closer to exams, just have a nice drink. Chill out for a minute. Hershey and Chase experiment. So Hershey and Chase experiment is a little bit tricky methodologically. So we're going to try to unpack it and make sure you're really clear on kind of the different components. So for the Hershey and Chase experiment, they used a virus to attack bacteria with the known belief that viruses infect bacteria or any organisms by transferring their heritable material. So the little virus is called a bacteriophage. A bacteriophage is a virus that infects bacteria. So it is not a bacteria, but it is a virus. And then they used E. coli as the bacteria. Please note this is not drawn to scale. So I'm just going to do a little not to scale because the bacteriophage is tiny compared to the E. coli. I need to be able to show different parts of it. So what is known is that this invades, so virus that invades bacteria, and it is known that it does that by inserting its heritable material. So the way viruses work, and we already understood this by this time, is they insert instructions and then the cell uses the instructions to make new virus parts. So whatever they insert has to be the genetic material. So then the question becomes, what do they insert? So if we can answer what they insert, then we can work out what the genetic material is. So it's made easier by the fact that the bacteriophage or the virus only contains 2 real parts. So it contains this external shell, includes kind of all these little spiky bits, and those are the protein. So So protein coat, and the protein coat contains sulfur. So we're gonna, and they use basically a radioactively labeled sulfur. And then inside of it, it has DNA. And the DNA contains phosphorus. And I'm going to do a pretty quick version of it because I don't want to spend 30 minutes on the Hershey Chase experiment. It's a bit complicated. So in terms of, they actually did it like 2 different trials. So first label this, see what happens, then label it. But we're just going to kind of join them together. So they're going to radioactive labels of both. Then they're going to basically give it a chance. So solution of viruses plus bacteria, which is going to give it a chance to invade. And then they're going to centrifuge it. And when you centrifuge something, what you're naturally going to get is the heavier thing is going to sink to the bottom and form what's called a pellet. And in this case, the bacteria cells are heavier. So the bacteria cells are going to sink to the bottom. So those are my pellet. And that's going to be the heavier invaded bacterial cells. And then at the top, then you're going to have the leftover kind of solution, and it's going to have the kind of like empty virus shells. So this is called the supernatant, which is going to be the leftover virus parts and the liquid. So that's kind of important, and again, we know that it only inserts the genetic material, so we're looking at which part is where. So you have to be kind of okay on pellet versus supernatant, and then what that means. So then when they tested it, what they found was they found phosphorus in the pellet, so that suggests that it is DNA that is inserted. They found sulfur remaining in the actual supernatant. So the fact that the actual invaded cells had the phosphorus that they had labeled in the virus, that's really important because they have their own phosphorus, but they have that radioactively labeled phosphorus there. That is important. So then what that tells us is that DNA was inserted by the virus. And then again, you have to come back to what we said up here. They only insert their genetic material. So that tells me that the DNA is the genetic material. So a little bit complex because you just have to be good on the elements and all of that, but that is basically what the Hershey and Chase experiment said. Okay, so that's kind of a little bit of an info there because I'm going to want to erase the board in a minute. So let's go from that. So that's saying, okay, yep, we now know that that is the heritable material. The 2 main forms that you need to know about are DNA and RNA. We did talk about in your notes that there are other forms. So ATP is actually an adapted nucleotide, but for right now, for this unit, you only really need to know DNA and RNA. So we'll get to ATP later, but it does fall under the category of nucleic acids. So for either one of these, let's make sure that we can draw one single nucleotide really clearly. Including kind of the differences between them. So we see that for both of them, they are made up of nucleotides. So that is a commonality between them. But let's look at how it's different. So it contains a pentose sugar. You can see me enjoying trying to draw that. So that is my pentose sugar. They both contain a phosphorus group, and you can draw it like this. You do not have to draw the chemical formula for things. And then they both contain a base. So I'm kind of drawing these intentionally to have it be like one's a pyrimidine and one's a purine. So we'll come back to that. I kind of used up more room than I intended, but that's all right. Here's my pentose sugar. It's supposed to look the same. It does not because I'm terrible at drawing, but it is intentionally looking the same. So I'll try to make it match it as best as I can with a nice big fat pentose sugar, and then a base like so, and then a phosphate. Okay, so far I've drawn them looking very similar. So let's clarify what the differences are between them. So for RNA, this sugar is called ribose. Let's number our carbons real quickly. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 holds up there. Carbon 1, carbon 2, carbon 3, 4, 5. So I'm just numbering the carbons. I don't use a number here because that's actually an oxygen. That's going to be important for directionality. So for HL, crucial. SL, you can probably get away with not knowing that super well. But for HL, you need to be able to kind of know what I'm meaning by that. At the 2 carbon spot for ribose, this is an actual OH group. Sorry, I'm writing that very small. So hopefully you can see that. That is an OH. I can erase this and make it a little bit bigger. So that has an OH there. So for the pentose sugar, that is an OH. For my deoxyribose, deoxy means one less oxygen. So it is actually just an H group. So this is deoxyribose. So they both have a pentose sugar, but it is different. The phosphate is the same. The base is what I want to look at next. So this base, I've actually drawn as a big base. So I'm going to call that adenine. So the bases that it is going to have is going to be A, G, C, and T. Meanwhile, for RNA, it has a unique base. So I'm going to draw this base here to be U. So it has the same other 3. So it also has A, G, C, but it does not have T. Instead, it has uracil. So uracil is the difference. And I'm going to go ahead and write that one out in full because I'm not going to write about it in the next little part. So there is my uracil. So it has one base that's different. While we're looking at the actual little nucleotides, and if you have space in between them, I kind of crammed it in, but if not, we'll just put it just below. So while we're looking at those, I want to point out that I did draw them as different sizes. So I drew this as a larger base. So the larger bases are called purines. And they consist of the 2 things. So it is one hexagon plus a pentagon. So hexagon plus pentagon. And those examples are going to be A and G. So that's going to be adenine plus guanine are going to be my purines. So those are my purines. And then I drew this one as one of the smaller bases. So that is an example of a smaller base. So the smaller bases are called pyrimidines. And they are just a 1 hexagon ring. So it's a single hexagon ring. And that's going to include thymine, cytosine, and uracil. So I always learn it as cuts aren't pure. So I'm going to say cytosine, and then uracil and thymine are going to be my examples there. So then if you remember cuts are not pure, that can help you remember that they are not purines. So cuts aren't pure because we have a cut, it's a source for infection, blah, blah, blah. That's a way of remembering it. Just a quick note that this material of the difference between purines and pyrimidines is HL only. So in order for base bonding to work, it always has to be between a purine and a pyrimidine. It's just an extra little word, vocab word for that. Okay, so what I want to do now, or we'll add our third difference and then we'll draw it. So then in terms of DNA versus RNA, DNA forms a double helix with two anti-parallel. I'm going to put that as kind of a key vocab term. So two anti-parallel strands that are bonded at the bases. With RNA, it does not form a true double helix. It doesn't form a double helix at all. mRNA is single-stranded, rRNA is single-stranded. The only exception is there's a small part of tRNA that does actually form some sort of base bonding. But in generally, we're just going to say generally is a small single strand. So tRNA is very small. So it does not form the double helix shape, which is an important difference. So where I want to go next is to actually draw that. So that's kind of my next, so that was about a quarter of my page. We've already done that. And then I want to draw them. So I am going to erase this just so I have the room to draw it. And so I'm going to erase everything and keep going. And draw kind of a double helix and a single strand and label the key parts in that. I'm going to go ahead and erase this as well so I have my whole space. Okay, so then we have DNA and RNA. Like so. Okay, so we are going to draw it now. I'm going to try to draw, we need to make sure you draw it with the anti-parallel, and I'm going to try to keep showing that pyrimine versus pyrimidine. You'll notice I'm going to try to draw like the hexagons. If you can't be bothered doing that, just make up a random shape. I don't think you really have to do that. I'm just doing it to make a point, particularly for HL. So if that's kind of annoying, just draw a random box is fine for the base, because we've already talked about that. All right, so I'm going to draw the double strand for DNA and the single strand. I'm doing 3 bases down. I probably could have even gotten away with only doing 2. So if you're running tight on space and you want to do that, you can. So I'm going to draw my phosphate, and then I'm going to draw my sugar, and then I'm going to draw a hexagon base, like so. Okay, then I'm going to draw another one, a phosphate, a sugar, and then this time I'll draw a hexagon and pentagon base. And again, you can just use a box if this is making you sad. I'm just trying to add another point to it. And then we'll just go ahead and draw 3, just because it looks more exciting than 2, but 2 would be fine. So feel free to just draw 2 on yours. And then I'll do this one. I'll do that again. Easier than drawing the complicated ones upside down. Okay, so there we go. So that's my start on it. I don't remember which colors I did last time. I probably should remember which ones I did for what, but I do not, so all good. So I will just make up and say this one I'm going to say is adenine. This one I will say is guanine. If you're using colors and you want to use the same ones you did on the last section, I just forgot which colors I used. Sorry. I think I might be close anyway. And then I'll make this one be thymine. Okay, so I'm just coloring those to represent that those are different. And I did deliberately draw my bigger purines and my smaller pyrimidine. Next thing I want to do is add my carbons. So this is coming off carbon 1, 2, 3, 4. The fifth holds the phosphate. 1, 2, 3, 4. The fifth connects the phosphate. It's kind of like somewhere holds up to the, creates the bond. 1, 2, 3, 4, 5 holds the phosphate. All right, all looks good. So now what I need to do, and I'm gonna actually go ahead and I'll draw the RNA as well. I'm actually gonna label them. I think I could do it. I just wanna make sure I have room for my other strand. I'm gonna draw all the DNA first, and then we'll draw the RNA. I'm gonna label it on the RNA side because I have more space there. So what I wanna go ahead and connect now are the actual backbone bonds. So this, to create a backbone, I'm going to create a bond here between the phosphate and the 3' sugar, the 3' end of the sugar. That is my phosphodiester bond. I'm going to label it on the RNA side just because I have other things to label here. Okay, now I want to show that this is double-stranded. So this T needs to have an A across from it. If my other strand was right side up, that wouldn't work. So the only way this is going to work is if this one is upside down. So here is my adenine that is going to bond with my thymine. It is attached to the sugar, and it is going to be upside down as a result. Here's my adenine. It is going to be attached to a thymine. It is attached to a sugar that is also upside down to accommodate that. And then this will be a cytosine, which is also a pentagon shape. So it will look the same as the one I just drew, like that. Whew, there's my upside down strand. So what you'll see by making sure I always pair a large and a small, the amount of space between the bases should be the same. So a couple things let's clarify. So we'll talk about complementary base pairing in a second, but because that is A, that means this has to be, or sorry, because that is T, that means this has to be A. So I can go ahead and label this as A. Because that is A, this has to be T. Because that is G, this has to be C. Okay? So now that I've drawn that, I can form, well, let's finish drawing connecting this strand. So again, 1, 2, 3, 4, and 5 holds the phosphate. 1, 2, 3, 4, and 5 holds the phosphate. 1, 2, 3, 4, and 5 holds the phosphate. So let's draw some nice phosphodiester bonds like so. So that has it connected at the sides, and then we can draw the hydrogen bonds, 2 between A and T, which technically you don't need to know in the new course, but surely it won't hurt you to know, and 3 between G and C. So there is my strand of DNA. If you have to draw it, you need to draw at least 4. You don't have to draw all 6 depending on your timing, but at least 4 are necessary to show everything. So this is my 5' to 3' strand. So a way of looking at that is your right side up strand. This strand is my 3' to 5' strand. And the fact that we have the 2 different ones suggests the term for that is anti-parallel. Another term you'll see for that relates to directionality. And that's going to be really important for replication, transcription, and translation. So this idea of here, so this is kind of the correct way to be. So 5' to 3' is used in transcription and translation. look a little bit more at that in a second, but that's just kind of a little note. So when we get to complementary base pairing, we'll pick that back up in just a second. Okay, so that is everything there. We are going to do a little bit on the complementary base pairs. I'm just going to, I don't know how much space you have. I'm going to try to move my RNA over a little bit because it's just going to be a single strand. So I am going to just kind of give one more little bit of information here. So I'm going to say hydrogen bonds form between complementary base pairs. So you need to be really good with that term. So anytime you're talking about kind of base pairing, you'll need to use that term complementary. And the complementary base pairs are always the same. So we'll write them here, and then below we'll talk about Chargaff's work and how we know that. But it should be pretty much clear from your diagram. So I'm not going to write out the full names again because we wrote them out earlier. So as long as you already wrote them, but do know that you need to actually know the full names. So you can't just write C and G all the time. Like you'll need to actually be able to say guanine and cytosine. So make sure you do practice that sometimes to be sure that you can actually do that. So then we'll say 2 base pairs here, 3 hydrogens, and there are 3. I like drawing them as dotted lines to show they're weaker. And then if you want to go ahead and put the arrow, but we're going to erase to actually explain it. So then we say, how do we know? And that's going to be Chargaff's work. So we'll kind of erase and go from that as our next kind of concept is how we know that. I want to finish with RNA though. So RNA is basically just going to look like this strand. So I'm just going to quickly draw that. Just because you could get asked to draw RNA instead. So just want to be clear, it should be the easy thing if you get asked for that because you don't have to draw any sort of other strands. So it's easier to do. So I'm just going to kind of quickly knock this out and then like so. Okay, so a couple things to note is we can label the numbers again because RNA does have a directionality as well. So 1, 2, 3, 4, and 5 holds the phosphate. It's probably, to be perfectly honest, less important on the RNA. So if you do not label your RNA, it wouldn't matter as much, but when we get to transcription and translation, it is nice. Let's make sure that we make one of these uracil, so we remember. So if you ever have to draw RNA, I would recommend that you draw one uracil, just because that makes it very clear that you know that. The other bases, they're not all uracil, so I'll still add a here, and then that would be a not a thymine, what's the other one? C. A C. Like so. Okay, and then the last thing that's really important is they need to be connected. So like I have here, so just like in DNA, we do have these backbone bonds. So I need to connect my phosphate to my 3' sugar. And then I want to make a little quick note of that. So those are going to be my backbone bonds. So they are found in RNA and DNA. Bonds, not bones. Sorry about that. Backbone bonds. So if we kind of summarize a couple things about them, we need to know that they are going to build the single strand of RNA, and they build both backbones of DNA. I want you to know a couple things about those bonds. First of all, they actually do have a name. They are called phosphodiester bonds, and you need to know that name. It basically means phosphate sugar bonds, so that should hopefully be pretty straightforward. So those are phosphodiester bonds. I need you to know that they are strong covalent bonds, so they are not going to be easily broken. I don't know that we labeled this here. Hopefully you remember this, but the hydrogen bonds are always weak and therefore easily broken. And then the last thing I want you to write now, but I'm not going to get into it because it's really a B1 principle, but once you're studying everything at the end, it's worth coming back to, that these bonds are actually formed by condensation reactions. So condensation reactions are any time we're kind of building monomers into polymers. So when you're studying for final exams and you know your condensation reaction stuff really well because you studied it in the rest of your biochem, that's a really good reminder that any backbone bonds are always formed by condensation reactions. Okay, so I should be at about slightly over halfway. So I'm going to erase and kind of keep going. The hard drawing part though is done, so that's hooray. And so like I said, I want to pick up with this. So how do we know about complementary base pairing? So how do we know that A goes with T and C goes with G? How did that come about? And that's because of the work of Erwin Chargaff. This is going to be, that last bit was all everybody. This is going to be a little like HL part only. I don't know if I wrote did last time, but do is probably a better way of saying. So how do we know specifically about complementary base pairing? And that's because of the work of Erwin Chargaff. So this is an HL only content. So we're going to say complementary base pairing was discovered by Erwin Chargaff. And where is the Hershey and Chase? We had to actually go into the methodology. You don't really need to worry about that as much here. It's really just the results. So we need to know the name, but you don't necessarily have to go into like the details of the paper chromatography and his methodology and so forth. I don't know why I felt like you need to know the word Erwin. You're probably fine if you just know Chargaff. So Chargaff made a bar graph, meaning he was comparing things. So the key thing that he found that's really important is that he found that the percent of adenine is approximately equal to the percent of thymine. So if you look at his actual data, it's kind of within the range of errors. So it should be the exact same, but paper chromatography, the way he did this, is not the world's most precise tool. So we're going to leave it as approximately equal to, but is definitely not equal to the amount of cytosine and guanine. So the percent of cytosine and guanine is always about the same. The percent of adenine and thymine are about the same, but they're not the same as each other. So kind of two important things. This actually, the fact that they're not the same, falsified the tetranucleotide hypothesis, which was that they're just a repeating sequence. So it would say that it would be like ATCG, 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 and if that was the case, they should have been the same. But that is not the case, but what he did find is he found what the pairs are. So he basically discovered these are the complementary base pairs. So that was the other important discovery. So a quick thing on kind of what is the implication of complementary base pairing. So like, why is it important? So what complementary base pairing tells me is that if I know one strand, I can predict the other strand. So if we say, what is the implication of this? So the implication is that each single strand can provide the template for the other strand. So each single strand can be a template, because if you know one strand, you know what the other strand has to be. And that's particularly important for replication, but also for transcription. So it's going to be really, really important for replication, because what it means is that we can split it apart and basically copy from one strand to make each new strand. So I'm going to just kind of do that as an example here. So if we had an original strand and we broke it open, and the original strand was A, T, C, G, A, G, and this strand was T, A, G, C, T, C. I could build a new strand off of that. So I could say, okay, if this is A, this one has to be T, this one has to be A, this one has to be G, this one has to be C, this has to be T, and this has to be C, and that would allow me to build a new strand, and then they would bond, and I've created a new strand, and then I could do the same here. So I could go A, T, C, G, A, G, cha-cha, and then draw some lines like that. So I used one strand as my template for both. Lots more on replication to come, so that's really just kind of a very overview, but just why it's important. Okay, so I'm going to keep going. I may erase though, just because I don't like having to squat down, but you should just keep going. But I am changing concept a little bit, and now looking at this idea of how does it provide the instructions. So we said that it is the instructions. Now the next question is like, how does DNA give instructions? And the answer is through what we call the genetic code. So we need to kind of pick up that little piece. So we kind of looked at structure of DNA. Now we need to kind of look a little bit at its function. So the genetic code is basically answering the question of how does DNA code for proteins. So we're going to kind of pack this down in the shortest little bit. So it's like literally a tiny bit, because this is going to be all of D1. So much more of this to come. So this is just kind of at this level, a little introduction to it. So if we say DNA is the instructions, and in particular, we're going to say DNA is divided into individual genes. So one gene is the instructions for one protein. One gene is one protein. Let's clarify, because we haven't done proteins yet if we're doing this in sequential order. So let's clarify what is a protein. A protein is a polymer, which is a fancy word for just basically a string of amino acids. So a protein is just a collection of amino acids in a particular order. And the order of those amino acids determines its function. So the order is really crucial. So I'm just going to make a little note of that the order of the amino acids is going to end up being the function of the protein. That's a really big point in B1. So more on that in other areas, but just for right now, what we need to look at. Okay, so we have our DNA. DNA actually cannot leave the nucleus. So if we're going to actually build our protein, that is going to actually be transcribed into mRNA. So transcribed or transcription into mRNA. Actually, I might change that to transcription just to make that term very clear. So you don't need to know any nitty-gritties about any of this in this topic, but I do want you to know the term mRNA, and then I do want you to know the connection between mRNA and DNA, and the transcription is the process that turns DNA into mRNA. Just that one line is fine for right now. So that is going to create mRNA, which remember is just the copy for that one gene. So that is the copy of the DNA for one gene. Okay, so then once we have our mRNA, the way our mRNA works is every 3 bases, particularly of the mRNA, is the code for one amino acid. So it is the codon, We're going to use that word a lot later, so we may as well use it now, but it's just the word for code. Codon for one specific amino acid. Okay, so a couple of terms I want to pick up here. So because every 3 bases, the genetic code is called a triplet code because it works in groups of 3. When we say for a specific amino acid, this is the same for every living organism. So the same 3 bases always code for the same amino acid in every living organism. So that includes bacteria, yeast, humans, monkeys, fungi, protists, plants, everything. So in every living organism, it is always the same code. So for instance, if we were to use the example, 3 bases, for instance, C, 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 always equals the amino acid proline. So much more on that to come. So for right now, you don't really need to know how that is built, but you do need to know that just term that it is universal. So we refer to that as the universal code. So in this part, that's really the key. Not that you understand how the mRNA codes for it, but simply that it is universal. Last little bit is the amino acid, just so that we've covered all of the different RNAs. I'm not going to do much on rRNA, but we are just going to make a note that the amino acid is delivered to the ribosome during this process that delivers it is called translation. So in this topic, we've just introduced the very basics of the processes. So we said transcription is making mRNA, translation is making the protein, and I can add that next little bit in if you want me to just clarify. Copy DNA into mRNA is the purpose of transcription. Translation is building the protein. And that actual delivery is done by another form of RNA, which is rRNA. Sorry, it's not. It is tRNA. rRNA makes up the ribosome. So that is transfer RNA, and it is involved in actually bringing over the amino acids. So mRNA is the copy, transfer RNA is going to bring over the actual amino acids. So really hard to get far into that because we need to go into more detail, which we will in D1. But for right now, you understand the universality, you understand the idea that the order of the letters matters. So basically what that means is that you can have very different base sequences on the DNA give different proteins. So different sequences and different links will enable us to create really different proteins. So the changes in the DNA is how we get to different proteins. So we need lots of millions of different proteins, but by having lots of different base sequences and lots of different links of DNA, you can follow the instructions and make really different things. Okay, one tiny little part, I'm literally gonna not even erase just to show you how tiny it is. The last little HL part only is we need to look at essentially if we have all this DNA, how do we organize it? So the HL part is gonna be on DNA packing. So basically the question is how do we organize a whole lot of DNA into the nucleus? So DNA packing is going to allow for efficient organization in the nucleus. So basically what it says is that don't just leave DNA out. So in my class, I always refer to it like thinking about like a binder and putting homework in a binder. So you got to organize your work in some way. Otherwise, if it's just everywhere, it's actually really hard to move and even a little bit hard to use. So you want some sort of organization system. So the organization system is going to be that we have these groups of 8 histones. So there's my 8 histones, which are proteins. So 8 histone proteins. Plus we have an extra kind of attachment site. This is called the H1 histone. So we have these group of 8 plus a kind of attachment site. And then my DNA wraps exactly twice around this. So here's my DNA, wraps once, and then we'll come back around and wrap a second time. So that's going to be DNA wrapped twice. There is a specific amount, so it is 146 base pairs. You do not have to know that exact number though, so I'm not going to write that down and just add more confusion. So we have 8 histone proteins, DNA wrapped twice, and then there's going to be a bit of a linker DNA, and then it'll be the next set. And then that'll be the next one. So we call that whole thing the 8 histone DNA plus this, plus that is a nucleosome. So this is really just a little vocab point. So a nucleosome is made up of the 8 histones plus the H1 histone plus the 2 types of DNA. So plus the DNA wrapped twice plus linker DNA. So all of that makes up a single nucleosome. So when we actually look at DNA, it looks like these kind of groups. So if we were to draw this, here's a little set. Not going to be super picky about drawing it to be exactly 8 because you get the idea. So when we observe DNA, it is actually going to look like that. So it's these little balls of nucleosomes with DNA linker in between them. And they will change. The linker can kind of move closer or further, so the DNA can be wrapped more or less tightly at different times. But for this unit, all you need to be able to do is define what a nucleosome is. Okay, that's a wrap. So well done and good luck.